Right. We will conclude this block of lectures for this test with signal transduction pathways. So what is a signal transduction pathway? This is how one cell sends information inside another cell. Okay, so you can imagine getting from one cell to a distant location for another cell, perhaps from the brain to the foot, or from the kidney to the liver, or something like that, where you can send a molecule, right? Send it through the bloodstream, send a signal from one cell to another. But once it gets to the target cell, how does that information get inside the cell? Generally, the molecule traveling is not soluble in lipids because it was soluble in water to get there in the first place, or the other way around, it was soluble in lipid and needed a carrier to get there, and now it can only get in the membrane, it can't get to the nucleus, it's got an ocean of cytoplasm in front of it. So we're going to talk about the ones where it gets to the outside of the membrane but can't get across. So instead of the molecule itself getting across, like the lipid membrane carrier or the lipid hormones do, here we're only going to send information across the membrane. So we're not going to send a physical molecule across. Right? So some terminology we'll use here are hormones that can't cross the membrane. I don't mean the lipid hormones, I mean other like protein-based hormones. We have neurotransmitters that work this way, growth factors, insulin, and other things like that. They can't get across the membrane, but they send information across the membrane. So we need something called a transducer. Right? And what the word transduce means is literally from Latin. Trans means across, and the verb ducere means to lead or guide. Right? And we get a word from this, and many of you are interested in becoming that. And the word ducere, we get the word doctor from. So doctor were originally guides or leaders, right? And physicians have since adopted the term to use as their own, but it also means leaders or guiders. So someone who is a doctor is educated in something and they lead or guide someone else to that information. So a transducer would be something that leads or guides across something else, right? So in our case, we're getting information across a membrane that cannot be bypassed by the molecule itself. And once that information gets inside the cell, we need to send it on its way. And in sending it on, we need some other messenger to carry that information. So there's usually some small molecule that's going to carry that information to another part inside the cell. A great example is a molecule that now goes on and activates another enzyme, or goes to the nucleus and acts as a transcription factor, or turns off a process in a cell. So one cell can tell another cell what to do. Okay, this is critical to be a multicellular organism. Okay, so let's look at some of the players in here. So in the top right, we have our original signal. Some cell releases a molecule. We call it the primary messenger. Right? That primary messenger, there's lots of these. We'll talk about a few of them. They go to a target cell or cells, and that cell receives the message. Right? It's the primary messenger binds to the outside of that cell. So there's a receptor that binds to that ligand. This signal could even be light. Right? And we receive the signal by binding or accepting the photon's energy. So we have some molecule receptor binding the primary messenger. That information then gets across the membrane. No molecules crossing the membrane, just the information is, right? And that's called transduction. And it usually is amplified in this process. And in the process of getting across, we transfer that information in the form of a new second messenger. Right? Not all of these cases will have a second messenger, but they often do. We'll talk about which ones do and which ones don't. But when you have a second messenger, that can become multiple copies of that second messenger. As long as the signal is still bound on the outside, we can make many, many copies of this second messenger. Right? So we have amplification at every step in the process. Once the first signal is received, the ligand binds to its receptor, we can get the information across, transduce it, and then turn on many copies of our second messenger. Send out the flyers to every part of the cell that wants to listen. Okay? There are only a few second messengers, and the ones we're going to talk about are drawn here on the screen. So one of them is called Cyclic AMP or Cyclic GMP, just depends which purine is attached. And you notice it's just a purine, A or G, attached to a ribose, and it has a phosphate on the 5' side. but it's bridged to the 3' side, right? So it's a 5' to 3' bridged phosphate. That's why it's called cyclic AMP or cyclic GMP, right? And it's made from ATP or GTP in the process. So we lose the latter two phosphates by having the 3' OH attack the primary or first phosphate. Okay. Another second messenger is simply a calcium ion. We talked about it briefly in our last lecture, but calcium ions could be released into the cytoplasm to facilitate muscle contractions. Right? So this calcium ion with its +2 charge isn't going to be floating around by itself, just like our sodium and potassium. It has a hydration shell of water molecules, so it's a lot bigger than it looks. We also have another molecule on the right called inositol triphosphate, or IP3. Right? And you remember inositol was our cyclohexane molecule. It looks like a sugar, but it's not a sugar. Right? It's a 6-membered ring of carbons. So it's a cyclohexane, and it's got an OH on every carbon. But in place of some of the OHs, we replaced them with phosphates. So on carbons 1, 4, and 5 around this ring, they've been replaced with phosphates. So it's got 3 phosphates on it. So it's called inositol triphosphate, or IP3. The other molecule we'll talk about is the one at the bottom. It's very simple, and you've already drawn this before. If you notice, there's a glycerol on the right? So 3 carbons here, OH on each carbon, and to these two, we've attached fatty acid chains. And if I were to put a phosphate on it, this would be my phosphatidate. But it's phosphatidate without the phosphate. So it's just a diacyl glycerol, not a triacyl glycerol. It's only got 2, so a diacyl glycerol. And that's one of our other second messengers. And these are the ones we're going to talk about when we get to the pathways. Eventually, as we're sending this signal along and the cell responds to it, whatever that response might be, to send out receptors onto the surface, to import glucose, to contract as a muscle cell, to turn on or off any particular gene, eventually we must stop doing that. There must be a termination of this. And that will end when the receptor is no longer receiving the signal. So the concentration of the ligand goes down, the light gets turned off, whatever the molecule is on the outside that's causing this thing to start is decreasing in concentration and going away, right? Eventually we'll turn all these systems off. We have to turn down all this amplification and eventually stop the response we had, but it's not instant. Okay? So to see this another way, here's an example. We have a membrane here on the page. We have some receptor in the membrane. The first messenger binds on the external side, right, the outer surface. Some examples of these might be insulin, a glucagon receptor, adrenaline, a photon of light, right? Lots of possibilities for our stimulus. Not a hormone, not a steroid hormone, because those will go right through the membrane. We're talking about protein hormones, for example, that will bind on the outside. Right? So that transduces the information across the membrane. We'll see several ways of doing that. And on the other side, on the cytoplasmic leaflet of this membrane, we have some effector enzyme. Right? So it might turn on a second messenger. It might bypass that and give you the response directly. We'll see 3 examples of how we can accomplish this. But there are very few second messengers. We just listed the ones on the previous page that we're going to talk about. Right? Steroids kind of bypass this process. They, instead of binding to a receptor that's facing the outside, they can pass through the membrane and bind to a receptor on the inside, and those can easily just migrate to the nucleus and have their effect. So that's not the pathway we're talking about. We already talked about that one when we did our lecture on transcription factors. Okay, so one more time, let's go through this process. We have a signal binding, a ligand binding to its receptor. That information goes across the membrane. That's called transduction, right? So it's been transduced, right? And every step along the way, we're going to amplify this. So I'm going to make a enzyme functional, perhaps, and it's going to make many copies of its substrate into product. It doesn't just do one. It does it over and over and over, right? And then each of those products might be an activated enzyme now, and they'll make their substrates into their products, which again might be an activated enzyme. You see, this is a cascade of events. And at every step along the way, there's amplification. So one molecule binding to its receptor, or perhaps 2 molecules binding to the receptor, depending on the type, can result in thousands to millions of molecules being activated inside the cell. Right? The technological Analogy to this would be a photomultiplier tube, if you know what that is. So they use them in Geiger counters, for instance. So it's a little cylinder tube where it detects some energetic particle on one end, and it only has to detect one. And then because this thing is powered, it sends a cascade of electrons down this tube. And every one of those that hits a plate sends another cascade of electrons. And every one of those that hits the next plate sends another cascade. So by the end, you have so many electrons moving that it creates enough current to make the audible beep you hear in these things, right? So it powers some speaker, right? So if you detect a lot of these things with your Geiger counter, it beeps rapidly. If there's just a few, it beeps less rapidly. So it's the same idea here. One signal will have an effect, one signal molecule, but if you have many more signal molecules, the response is drastically improved. It's orders of magnitude higher, not just double. Okay? The three types of receptors we're going to talk about, and we'll spend probably most of the time on the first one. They're called 7-transmembrane helix receptors, and you've already seen an example of this in rhodopsin or bacterial rhodopsin in last lecture. And there's 7 transmembrane alpha helices that make up the bulk of the protein. The second type are called dimeric receptors that recruit kinases. Right? And we'll see an example of that in growth hormone receptor. And the third type are again dimeric receptors, but they don't recruit kinases. They themselves are kinases. It's kind of the all-in-one receptor. And an example of that is the epidermal growth factor. Okay, so let's look at these three types. And the first one you've already seen, the 7-transmembrane type, where you have 7 times this protein crosses the membrane. So one side will end up on the inner side of the protein, inner side of the membrane, and one side on the outer side of the protein. Okay. These 7 alpha helices don't line up like this. You know, they fold into a cluster or a bundle. And these type of receptors are used for all of these functions you see on the right. So it's most of our functions. And in fact, you should point out that 3 of them in particular are used by this type of receptor, right? That's 3 of what we called your 5 senses. Right? Which 2 are not represented by this type of receptor?
Hearing and touch.
And how are those detected? So clearly, smell, taste, and vision are detecting a ligand or a molecule or light. How are hearing and touch receptors? How do they work? They both work on the same principle.
Is it? I know with hearing, it's like mechanical stimulation.
That's right. Same thing with touch.
It's like the little drum in your ear, and it actually mechanically moves and causes your neurons to be stimulated through.
Well, I'm not looking for the details of how hearing works, but how are hearing and touch similar? What are they detecting? Like these 3 are detecting molecules or light. What are hearing and touch detecting?
Pressure.
Pressure, right? They're both forms of pressure. So in hearing, it's a series of pressure waves. And in touch, you're physically touching it, right? You're moving it, right? It's detecting the pressure put on that receptor. So those don't use these 7 membrane type receptors, but you notice the other 3 do, along with many other functions in your body. Right? All these use a 7 transmembrane type receptor. It's by far the most common. Okay, so let's see how it works. We're going to use one particular one as an example. It's the classic example of the beta-adrenergic receptor. And it's a receptor found on almost every cell type, and it reacts to a beta-adrenergic type molecule like adrenaline, also called epinephrine. Those are the exact same thing. One's in Greek, one's in Latin. Right? It's adrenaline is made by the adrenal gland, which is the gland on top of the kidney. Renal means kidney. Adrenal, which means on top of the kidney. So it's the adrenal adrenaline. Epinephrine, nephrin is the functional unit of the kidney, also referring to kidney, and epi again means on top of. So on top of the kidney molecule is epinephrine. They're exactly the same thing in 2 different languages. Okay. If you had to guess where epinephrine or adrenaline is derived from, what molecule do you think it's made from based on its structure there? It's very close to something you're familiar with.
Is it cholesterol?
It's not cholesterol. There aren't 4 fused rings It's much closer to the actual molecule it's made from than that. Right? I'll give you a hint. You see these three OH groups. What if these two, the top two, were not present? Only the one on the end were present. And attached over here, where this methyl group is, let's lose the methyl group, and in place of it, next to the nitrogen on the other carbon, we put a carboxyl group. Right? What would it be then? So no methyl group, and in where the nitrogen is, next to it put a carboxyl group on the other side, and lose those other two OHs.
Would it be a tyrosine?
That would be tyrosine. So this is made from tyrosine, right? So, and you can probably guess the steps involved. We have to decarboxylate it, lose the carboxyl group. We have to methylate it, and we must put hydroxy groups on it, 2 of them. And then you have epinephrine, right? That's how adrenaline is made, right? So what this molecule does is it's released by the adrenal gland and goes all over the body, right? It's receptors on almost every cell type. And it tells those cells to react in a certain way, right? So if epinephrine is released, what does it do to certain? What features do you know about this? It's responsible for what they call the fight or flight syndrome. But what does it do to you? What do you experience?
Increased heart rate.
Heart rate goes up, yes. What else? So besides the heart rate going up, anything else to do with the blood?
Blood pressure.
Pressure goes up because this molecule tells those muscle cells surrounding arteries to contract. So the blood pressure goes up quickly, right? And it binds to a different cell type and in fact tells those cells to relax. Has the exact opposite effect. Where are those?
Your digestive tract.
Well, it does shut down the digestive tract, meaning it doesn't let it continue the slow digestion or compression there, but I'm thinking more immediate. Where you'd like vessels to open, not contract.
Your airway?
Right, so it stimulates the opening of the airway, so the lining of those are told to relax, not contract. So the same molecule can have the opposite effect on 2 different cell types, and that's because they differ not in their receptor, but in the downstream effects it causes. So inside the cell, once we get it through the receptor, we transduce it, we get the information across, what those cells do with that information is quite different. Okay? We're only going to talk about how it gets across, but different cell types can have different effects from the same ligand. That's what I want to make a point of. Okay? Here's another example of a 7-transmembrane receptor called rhodopsin. Right? This is just like the one you saw before, which was called bacterial rhodopsin. This one's just rhodopsin. It's not the bacterial version. This is what's found in your retina. And in the center of this cluster of alpha helices is the ligand that's permanently bound, right? And this is your vitamin A, retinol or retinoic acid molecule with its long fatty acid tail and single double bond in it. And when light hits this, it changes its shape from being trans to cis or cis to trans. And when it changes shape, then these helices have to adjust around it. And when the helices have to adjust around this molecule that's changing shape from being bent to being more straight, being cis to trans or trans to cis, the loops on the cytoplasmic side have to reorient themselves as well. And those changing loops on that side are what sends the signal on. So this one has a permanently bound ligand, the retinol molecule, but it changes its shape depending on light. So this is what allows you to detect light. So when a photon of light hits this, it changes its shape, the cytoplasmic loops readjust, and the cell knows there's been a change somewhere, right? This molecule has had to readjust, and it sends a signal to the rest of the cell. Okay? We'll talk about what that signal is next. Okay? So it's not just one thing. This process can be used to detect lots of things, including light or other molecules. But once it gets to the inside of the cell, we have a very common thread for all of these. They follow very similar pathways. Right? And these are called G proteins. not because they have their own magazine or anything or they're cool, but they're named because they have a GTPase activity, right? So in other words, they're proteins that have the enzymatic activity to hydrolyze a GTP molecule into GDP and phosphate, right? They don't phosphorylate anything. They're not kinases, but they just have the ability to hydrolyze that bond. And they'll only do so if they're arranged correctly or told to do it, right? You'll see what I mean by that in a second. Okay? So almost all of these G proteins have the ability to hydrolyze GTP, but they do it slowly. And when I say slowly, I'm referring to the infrequent ability to do it. So if a G protein has GTP bound to it, eventually it will break that bond. It'll hydrolyze it into the GDP and the phosphate. But it doesn't do it very often. It's not very efficient at it, and that's the point. We want it to stay as GTP bound for a while, right? We're talking seconds, right? Maybe a few seconds, right? Most a minute, but not forever, okay? And eventually it will hydrolyze it, and it'll be a GDP bound again. And if I want to do that again, I'm going to have to remove that product, the GDP, which it doesn't want to let go, and replace it with a new GTP. And this is where this thing comes, is where the receptor comes in. Okay? So at the figure at the top here, let's use the epinephrine receptor as an example. Epinephrine binds to its beta-adrenergic receptor. And in this diagram, it's somehow glowing, right? That's their indication that it's bound, okay? Normally, these G proteins, right, the group of proteins underneath is called the G protein. It has 3 parts. It's a quaternary structure of 1 alpha, 1 beta, and 1 gamma, right? So it's a heterotrimeric structure, 1 alpha, 1 beta, and 1 gamma. And relatively speaking, these are about their masses. The alpha subunit's the biggest, the beta's a little smaller, and the gamma's very small. It's essentially a pair of alpha helices that are wrapped around beta. And if you look at the actual structure on the bottom left, you'll see it mirrors that assignment. Okay, so the alpha subunit is shown in like the almost white with the purple background here. The beta subunit in blue, I matched it to the color here, and then the yellow gamma subunit sitting around the outside. The beta and gamma subunits, you can think of them as a group that stays together. Beta and gamma stay together all the time. When this group is sitting there in the cell's been relaxing, doing nothing. It has a GDP bound to it because at some point in the past it hydrolyzed the GTP and it's been sitting this way ever since. Okay? It finds a beta-adrenergic receptor up here that has been activated by its ligand. So epinephrine binds to it, this thing changes shape slightly, and now this G protein can bind to this thing. And it basically acts like a can opener, right? It can remove the beta and gamma hat, right? The beta and gamma group can be removed, leaving just a naked alpha subunit. Because the beta and gamma were there, the GDP could not leave. As long as beta and gamma were attached, GDP could not leave. But now that this receptor with its bound ligand is a functional can opener, it takes the beta and gamma subunit off. GDP could leave now, and it's replaced by a GTP. Right? With GTP bound, the beta and gamma subunit cannot rebind. They can only rebind if it hydrolyzes it. So temporarily, it's safe from its hat, from the beta and gamma subunit cap coming back on. But while GTP is bound, this alpha subunit has the ability to bind to this other enzyme called adenylate cyclase. And I'll show it on the next slide how this works, and the interaction here is appropriate. So because this alpha subunit has lost this piece, the beta and gamma subunit, they were getting in the way of this interaction before. But with GTP bound, right, it can interact with adenylate cyclase and not with its beta and gamma subunits. Okay? We'll talk about what adenylate cyclase does in a second, but eventually our G protein at some point will hydrolyze the GTP. It's just bound to happen. It's a ticking bomb, and eventually will hydrolyze it, okay, at some point. When it does so, it can no longer associate with the adenylate cyclase, and the hat, the beta and gamma subunits, will once again bind. But what if the epinephrine is still there? It's in such high concentration that it's still activating this receptor. Then it can come over and get its hat removed again. As long as epinephrine is still bound, the can opener still works. Right? And it'll eventually replace the GDP with another GTP, and we'll repeat this process over and over and over. And as long as GTP keeps getting replenished in there, I can keep activating adenylate cyclase. Now, each beta adrenergic receptor can do far more than one of these, right? Everyone that comes by can get its beta gamma subunits removed. So it'll activate thousands of these G proteins. And each of those activated G proteins will activate many adenylate cyclases. You see the amplification we have going on here. What does adenylate cyclase do? Well, like the name implies, it takes an adenylate, like ATP, and makes the cyclic form. So it takes ATP, cuts off the beta and gamma phosphates, right? Beta gamma leaving here, beta gamma leaving here. And makes the cyclic AMP with its alpha phosphate. And that's what we showed on the previous slide, where the 5' phosphate is now also attached to the 3'. Okay? In that form, cyclic AMP activates another enzyme called protein kinase A. And we'll see that on a future slide. Okay? So here's my G protein again, the alpha subunit. One more time, I'm going to flip back and forth so you guys can see that, right? So in this slide, here's the alpha subunit one more time, and you see the beta gamma subunit is sitting right where it would normally interact with adenylate cyclase. It's either one or the other. It's either bound to the beta gamma subunit, and it does so if there's GDP bound. But if this gets removed and I replace the GDP with a GTP, like this, it can now associate with adenylate cyclase. And this part of adenylate cyclase is the soluble part of it, the part sticking out into the cytoplasm, right, where it has all these helices in the membrane. It is not a 7-terminus membrane receptor, but it definitely is an integral membrane protein, right? It's this piece back here, right? I tried to color code it for you. So it'll bind to the adenylate cyclase right here, making contact with these two fragments, and that activates this adenylate cyclase. And what it does is turn ATP into cyclic AMP. All right? So cyclic AMP has been created here. Cyclic AMP has the role, and of course this enzyme will make many of these. It doesn't just do one, it makes thousands to millions of them again. It's a very rapid enzyme. The cyclic AMP goes and binds to protein kinase A. Here's a diagram of protein kinase A at the bottom. It has two parts. It has a catalytic subunit in blue, the functional enzyme. It has a regulatory domain in yellow here, labeled R. Normally they're bound to each other in this region. And as long as these are bound together, this does not function as a kinase. It's inactive. In order to get these two to separate, I need this yellow regulatory domain to bind 4 cyclic AMPs. And when it binds these cyclic AMPs, which are not normally around, but they are now, they separate the regulatory domains from the catalytic domains. These catalytic domains then act as a functional enzyme. They're protein kinases. What do kinases do? They transfer phosphates. They take it from one thing and give it to another. The most common example is to take it from an ATP and give it to a protein, right? And that's what this guy does. And it doesn't do it to just any specific protein. It has a variety of targets. That's why it's just called protein kinase A. It's one of several kinases. And this one phosphorylates on a number of targets. At one point in the past, I made a meme about this. And it was, do you guys remember the Oprah Winfrey show where she gave away a card to everyone in the audience? That's kind of what these kinases do, right? So if they see a target, it gets a phosphate, right? So this protein kinase A phosphorylates many, many targets, right? So everybody gets a phosphate. So will this go on forever? No, we need to stop at some point. How will we shut this whole thing down? Eventually, we must remove the epinephrine. Right, so on the outside of the cell, it doesn't bind permanently to the receptor. It binds and releases all the time. But if the concentration stays high enough, it stays bound more often than released. But if the concentration finally drops, which it eventually should, right, this beta adrenergic receptor will stop glowing in this diagram and will no longer be active. At that point, any G protein that is hydrolyzing its GTP and coming back to get it replaced will not happen. Once the beta and gamma subunits go back on, it's out of luck. The can opener is not working anymore. I can't remove these, and I'll no longer activate the G protein. But that takes time for all those to hydrolyze their GTPs. Remember we said several minutes perhaps. And then the GTP bound ones that are still around will eventually hydrolyze theirs, and they'll all be back in this form. Eventually, that means my adenylate cyclase is no longer making any more cyclic AMP, but there's plenty still around. So it takes a while, but cyclic AMP is inherently unstable. That bridging phosphate between the 3' and 5' carbons will eventually break, and we'll end up with just AMP. Right? And AMP, when it does finally break, doesn't fit in this pocket very well, and it dissociates from the regulatory subunit. Once enough of those have dissociated, the catalytic subunits rebind to the regulatory ones, and this thing stops putting phosphates on things. But all those proteins that it phosphorylated are still going to be active. Eventually, we must remove all those phosphates. So this also takes time. So this is why if you have a burst of adrenaline or epinephrine released, you're driving along on the highway and you almost get run over by a tractor trailer, it isn't over immediately late after that. You're going to have to pull over and listen to your heartbeat for 5 minutes, because it takes time for this very rapid accumulation of activated things to turn off. Okay. So here's an example of that Gprotein cycle. I have my activated G protein, just the alpha subunit. It's activating adenylate cyclase by touching it here and binding to it. Eventually, it will hydrolyze its GTP. I just have plain old GDP. That does not activate this thing, but it does make the appropriate shape so my beta and gamma subunits can reattach. And as long as they're reattached, I can't activate this anymore. And since my epinephrine has dissociated, right, this thing's no longer glowing, right, the bottom looks a little different and it can't remove these anymore. Right, so eventually we downgrade everything. Of course, I already said the cyclic AMP is transient. It's not permanent. It eventually hydrolyzes itself with water. It's not stable. It just turns into AMP, and the signal will stop growing at that point. But all our proteins still need to be dephosphorylated, and a protein phosphatase will come along and do that. Okay? So how do we actually hydrolyze GTP? So here's a molecule of GTP, right? And in the active site of our G protein, we said it does this infrequently, right? It cuts off this last phosphate, right? What if you had a protein, a G protein, that could not do this? It could not hydrolyze this GTP, right? So what would happen? Well, it would, go back a second, it would get activated here at the receptor. The beta and gamma subunit would come off. It would drop its GDP, pick up a GTP, become active, turn on adenylate cyclase. And now we have a problem. It'll never hydrolyze its GTP in this scenario. Well, that means I'll never turn this signal off. And ultimately, I'm just going to waste all my ATP, turn it into cyclic AMP, and phosphorylate everything. So it's like you can never turn the signal off. Even if the epinephrine leaves, I can't get this out of its active form. And this is what happens with cholera toxin. It's a protein that binds to my GTP-bound G protein alpha subunit. That's a mouthful, but it's this piece with its GTP bound. And the cholera toxin protein binds to that and prevents it from doing this reaction ever. Okay? So we just completely hydrolyze all our GTPs and get it back to normal. But in this case, we can't get back to normal. Right? We're constantly making cyclic AMP. And so eventually we lose lots of water. We hydrolyze all our ATP. We're going to lose salt and water. We're going to urinate all this out. And then we go back to the water fountain because we're thirsty now. And that's exactly where the cholera toxin is. Right? So we go back and get more water. We further dehydrate ourselves. We go get more water. This is a downward spiral. Right? And that's why this was called death's dispensary. If you ever get cholera in the water supply, it's just a never-ending cycle and people die of dehydration. Right? So you should check the central water supply, make sure there's no cholera in it before everyone starts dying of dehydration. This was a problem back before we had all the central water and filtering and things like that. Okay? The whooping cough causes exactly the opposite thing. It allows it to hydrolyze the GTP instantly. So we never get the signal. Right? So you can release all the epinephrine you want, and your cells never get the signal. That's the opposite cause there. Okay? So some other potential processes, we have other second messengers besides these cyclic AMPs. We have one that activates a protein called protein kinase C, not A. So if you imagine this is a Sesame Street slide, everything on this slide is brought to you by the letter C. Our ultimate goal here is to activate protein kinase C. It's another kinase. It's not protein kinase A this time. It has a different set of targets. Maybe this is a different talk show, right? They give away tractors instead of cars. I don't know. But it's a protein kinase. It still gives away phosphates. It puts them on things. It just has a different set of targets. Okay? So what happens here is if we have our activation by protein kinase A from the previous slide, and that activated a lot of proteins, one of the proteins it activated was this one called phospholipase C. Right? It's up here at the top. Phospholipase C. And it is a lipase. That means it cleaves lipids. It cleaves phospholipids. And here's our phospholipid that it cleaves. It's called PIP2, which stands for phosphatidylinositol, which you should recognize. It's a glycerol, 2 fatty acid tails, a phosphate, and an inositol. This was phosphatidate with an inositol on it. Right? So this is one of the ones I had you know. And on the inositol are a pair of phosphates. So it's a phosphatidate with an inositol on it and 2 additional phosphates. That's why it's called phosphatidylinositol di or bisphosphate. Right? So this one phosphate's bridging the 2, and the other 2 are on the inositol. That's the reason behind the name. What this phospholipase does is it cleaves this bond right here. Between the black and the red parts here, it cleaves this bond. So it's cutting off the remaining piece here, the diacylglycerol, which is just the glycerol and the 2 tails left, and the inositol, which it looks like it's gained a new phosphate. It's called IP3. It's inositol triphosphate now. But it's simply the 2 it had plus the one that was bridging the connection have all left together. Okay, it didn't gain a new phosphate. It was already there. Right? But I'm just cutting it off at this point. So this phospholipase C now creates 2 second messengers. This thing was just sitting in the membrane minding its own business when phospholipase C cleaves it into 2 pieces. A diacylglycerol, DAG, and a inositol triphosphate, or IP3. That's shown diagrammatically down here. So we have our PIP2 sitting in the membrane here. So here's our 2 tails, right? And this is the glycerol. Here's the phosphate bridging it. And then the blue hexagon represents the inositol, and then the 2 more circles represent the additional phosphates. Okay, so phospholipase C cleaves this bond again, right? Shown here in the diagram, and I get 2 products. Diacylglycerol, which is clearly not soluble in water, stays in the membrane. And then inositol triphosphate, which is highly soluble in water with all these phosphates and OHs, leaves into the cytoplasm. Okay? What these 2 things do, let's follow the inositol triphosphate first. It binds to a receptor on the ER membrane, right? So this first one was in the cell membrane. This is in the ER now, or in the case of a muscle cell, it's the SR, sarcoplasmic reticulum. There's a receptor there for IP3. Normally, IP3 doesn't exist in the cytoplasm unless this enzyme has been activated. So we release the IP3, it floats over here, binds to this protein. This protein receptor is also one of our channels we talked about in our last lecture, and it's a channel for calcium. So while these are bound, right, the gates open and let the calcium rush into the cytoplasm. It's normally in high concentration inside the ER or SR and lower concentration in the cytoplasm, so it rushes out. And now calcium is acting as my messenger. So I have my calcium messenger and my diacylglycerol from before. They both bind. Since protein kinase C is next to the membrane here, it binds the diacylglycerol. It also binds calcium. If I get both of those signals, protein kinase C is now active. So it's another chain of events where you lead to activation of an enzyme. How do we turn this off? Eventually there's a pump, not shown in the figure here, that pumps those calcium ions back into the ER, right? And our IP3 eventually gets cleaved. We lose a phosphate off of it. It's no longer activating this enzyme. And eventually my phospholipase C loses its activation and doesn't create any more IP3s. Right? So eventually the whole process is set to turn itself off. Okay. The second class of receptors we'll look at are the dimeric receptors that recruit kinases. Okay, these are not 7 transmembrane things at all. Okay, so this works quite differently. So imagine, let's do the diagram here first. So you have a cell membrane, and in it are the receptors we're talking about, the dimeric receptors that recruit kinases. So here we have two identical pieces, and some ligand, we'll use a growth hormone as an example, comes and binds to one of them, shown in this picture. Doesn't do anything in and of itself. But since it also has a site on the other side of the growth hormone, as shown in the picture here, it's a molecule that can bind many of these things, right? It binds at least two of them. If you bind two of them, if they get close together on the outside of the membrane, then they must also be close together on the inside. And normally they would never stay together on the inside. They float around randomly, the lateral walk. But if this ligand holds them close together, and these two stay close together, we call it activated. Because these two pieces are in proximity, they form a binding site for a kinase. Much like our protein kinase A, it's just a different kinase, it's not protein kinase A. It binds to this dimerized tail of my receptor. So if we look at its name again, it's a dimeric receptor, meaning I need the two pieces put together, and it recruits or provides a binding pocket for another kinase. It is not a kinase itself. Okay, so an example here is one cell releases a growth hormone. It binds to other cells, and those cells dimerize the receptor. A tyrosine kinase binds to this, and that kinase gets activated. Right? So an example of that is our Janus kinase at the bottom. Right? And that kinase does what kinases do. It phosphorylates a large number of targets, not necessarily the same targets as the ones we've seen, and those regulate gene expression. They act as transcription factors to turn on certain things, like turn on the cell cycle enzymes to start growing and dividing. Okay? The third class is almost exactly like that, except it says we're going to cut out the middleman. I don't need the help of that. So here we have our membrane receptor again, another copy of it here. And this time when our epidermal growth factor, for instance, right, binds, these two bind to one another, and because they don't recruit a kinase, they are kinases in and of themselves, they autophosphorylate each other. So one will phosphorylate the other, and I get phosphate stuck on them. An example is our JAK kinase. Right? When they phosphorylate each other, then they're activated. And then these two things, being kinases, phosphorylate a subset of other things. Right? So unlike the second class, they don't recruit a kinase, they are kinases. They cut out the middleman. They do the job themselves. Okay? An example of that is our epidermal growth factor. So here you can tell a cell to start dividing, but only with this type of receptor, and it's only found on epidermal tissue. Right? It's not going to be found on nerve cells because you don't want those dividing. Okay? So here's that epidermal growth factor pathway in all its glory. So we have our epidermal growth factor binding to a pair of receptors. They dimerize, they autophosphorylate one another, and of course, being kinases, they phosphorylate something else. And downstream of that, depending on the cell type, they could phosphorylate a number of other things. Activate transcription factors, even activate other G proteins. But what I want you to remember here is this G protein is not a result of this transduction. It's a downstream event, much farther down. Right? This is not my typical G protein that gets activated here. So the epidermal growth factor, or the type 3 receptor here, does not use a second messenger directly. It could have one downstream, of course. All of these things can. But it doesn't use one directly. Whereas in the first case, back here, there it goes. We did use a second messenger directly. Right? It was binding to the protein or kinase receptor. Here, our second and third class may or may not use, well, second class may use a second messenger, but the third class does not use one directly. That's what I want you to know. Another example of this third class receptor would be insulin. Insulin binding to its receptor. I don't have a picture of it here for you, but insulin binding to its receptor will cause the same type of thing, but not activating RAS protein in this case. We're not trying to divide. It's going to activate a molecule that's sitting in the cell. It's a protein. It's sitting in the cell, and it's transported to the cell surface. And that protein we just transported to the cell surface was my importer that says, I'll take a sodium across if a glucose comes across with me. Right? That was my sodium glucose symporter. It's only going to be found on the surface in abundance if you have insulin bind and have this cascade of events that says you need to go to the surface. There's glucose out there, go get it. So that's how In a type 1 diabetic who doesn't make insulin, it's ready to go, it just never receives the signal. In type 2 diabetics, the signal is there, but some process in the pathway, maybe the receptor for insulin doesn't work, maybe the importer for the sodium glucose importer, symporter doesn't work, something along the pathway after the insulin doesn't work for the type 2s. Okay. So these are called receptor tyrosine kinases because in all these cases, these kinases are putting phosphates on tyrosines. Okay? We have three main targets for kinases: serine, threonine, and tyrosine, the three alcohol amino acids. Serine and threonine are generally reserved for activating and deactivating proteins and enzymes. Okay? 90% of the time it's going to be serine, 10% of the time threonine, but almost never tyrosine. Whereas in cell signaling, it's almost 100% tyrosine. Clearly looks a lot different from the other two. It's got the big benzene ring in it. So tyrosine is almost always the target for our receptors in cell signaling, so we call them receptor tyrosine kinases. Whereas activation or deactivation through phosphorylation of enzymes is almost always serine and occasionally threonine. And so these small G proteins we've been talking about, these monomeric G proteins, right, are used to send down this pathway. These guys down here, these G proteins, these RAS proteins, right, are activated by GDP bound or GTP bound in much the same way our other G protein second messenger was, and they're used to control our cell cycle, as you see here. All these things are involved in cell cycling. So if you mess up this tyrosine receptor kinase for these epidermal growth factors and suddenly this RAS protein is always on, you're going to get a tumor cell. It's going to keep dividing even when it's not supposed to. So much like our cholera toxin, we're told it to always keep making the cyclic AMP, we have here keep being told to divide. And it's usually a problem with the tyrosine receptor kinase or the cytoskeleton protein or the RAS protein or something like that. It keeps telling it to do this when it shouldn't. It doesn't get turned off, much like our cholera toxin caused it to never turn off. Okay. Here's the example of insulin. So insulin being the molecule at the top, it's got the two chains held together by the disulfide bonds. And on the right, we have our picture of insulin, and it binds in this little pocket. It's again our third type, a tyrosine receptor kinase, where if it binds, they dimerize. Inside the cell, they also dimerize for the beta subunit, and these things phosphorylate one another. Again, it's the all-in-one. There's no recruitment of a kinase. It is a kinase. So they cross-phosphorylate or autophosphorylate one another, and that sends a signal to my molecule, that's the sodium glucose symporter, go to the surface, embed yourself in the membrane, and import the glucose that's out there. Okay, so that's what's shown here. We have our binding, phosphorylate each other. We phosphorylate other things. It's a cascade of events again. We employ our PIP2 turning into IP3 and so forth. And eventually activate an enzyme called AKT. And that enzyme, which you don't have to remember this whole cascade, I just want you to know the goal, is to put the glucose sodium symporter into the membrane so we import glucose. If any one of these parts doesn't work, that's a type 2 diabetic. Right? So lots of places for this thing to not work. So don't think type 2 diabetics are simply, the receptor doesn't work. It could be any one of these functional parts not working, and you don't get the signal to affect transmission of information. If you don't make the insulin, however, that's type 1. Okay, so here's our process one more time. You see there's lots of parts. I don't want you to memorize the parts. I want you to know that insulin uses a tyrosine receptor kinase, the type 3, right? Every step along the way, there's amplification. So we get all of the available glucose sodium symporters put on the membrane. So rapidly glucose is imported into the cell. And that's how when insulin is released, we suddenly get a rapid drop in the blood glucose concentration, because every cell that has a receptor for insulin is suddenly importing glucose by this mechanism. It's very rapid. So how do we stop all this? all these things that are getting phosphorylated need to eventually have their phosphates removed. And we do that with phosphatases. So phosphatases take phosphates off, kinases put them on. So we have protein phosphatases to remove them off of proteins, and we have lipid phosphatases to remove them off of lipids. So, and these are always active. These phosphatases are always around. They're always constantly removing the phosphates. If we want to do this, we keep putting them back on. As long as the signal's there, we'll keep doing it. Yes, this is expensive, but cell needs to live. And lastly, we have Calmodulin, which is another molecule in the cell that when it binds calcium, it adopts a new shape. Okay, so it simply changes shape, and it looks like this EF hand, they call it, because it's a hand with your finger and thumb out. It's like you're counting to 2. So if calcium binds to it, it changes shape, and other proteins that recognize this new shape become active, such as Calmodulin kinase. Right, so it's a kinase again. We have a variety of kinases as usual, has a whole different subset of targets, and it only becomes active if this molecule, Calmodulin, binds to it, which it can only bind to it if it's bound to calcium. So this is a way of sensing the concentration of calcium in the cell. Okay, some problems with this is in CML. If you have part of your chromosome 9 and 22, they switch places. Right, so they look similar to each other, and these two things switch places, which means our epidermal growth factor, right, is now sensitive to something it shouldn't be sensitive to. So it's not sensing epidermal growth factor receptor is not looking for epidermal growth factor anymore. It's looking for a different ligand. So now a signal that should be harmless is telling a cell to divide. Well, that's going to generate a cancer cell, and because it's in the leukemias, or the white blood cells, it transmits it everywhere in the body because those are mobile cells. So what's happening is normal epidermal growth factor receptor, which is looking for its normal ligand of epidermal growth factor, because of this translocation of some of the DNA, it's still a receptor, but it's detecting a different ligand this time. And because it detects it all the time, right, whatever the ligand might be, because of this rearrangement, it's telling the cell to divide all the time. So that's why this white blood cell nonstop divides, and that's a leukemia. So if I can inhibit some of these kinases very selectively, I may be able to treat these types of cancers. But be careful, because these kinases have a large number of targets, so there may be many side effects. That's what makes this so difficult.